Hey Geeks, welcome to another video. So today we will be discussing about sequence similarity searches. This is the fourth part of my bioinformatics series, and this is a very important topic with respect to the DBT JRF syllabus. Now you won't believe it, around 75% of questions asked from bioinformatics belong to the topic that I'm going to discuss in the coming 3 videos. So please do not forget to watch all of these videos before going for your exam. Now, without much further ado, let's get right into this video. So first of all, as the name suggests, what are we going to do? Actually, we are going to do a sequence similarity search. That is, as the name itself tells you, we are going to search the similarity between sequences. Now, the similarity between sequences can be easily determined by different methods. Now, depending on whether the sequence is a nucleic acid sequence or a protein sequence, you can use different software. What is the aim of actually carrying out sequence similarity search? See, if you know that two sequences are very similar to each other, there is a chance that they may work the same way. They may have the same structure or the same function. That gives you better idea about the new molecule, right? So therefore, people go on to do sequence searching. Now, before even diving on to do the different methods, we have to understand some terms associated with this. So the first term that we have to understand is about the sequence similarity, and second is sequence homology. Many people interchangeably use similarity and homology. Now we are going to understand why that is incorrect. So let us consider there are two sequences, sequence 1 and sequence 2. Let's say sequence 1 is AAPT, CCGG, AA. Okay. So how many residues? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay. So we have a total of 10 residues. Now let's consider another sequence, which is from say another organism, which is ACC, TCC, A, T, T, A. Okay. Let's say this is the sequence. Now, what is the similarity between these two sequences? How will you calculate that? Very simple. Just see which bases are the same and you mark them. So now in our case, tell me which are the similar sequences here. AA is similar. Then we have TT that are same. Then C, C. Here nothing else. Okay, another AA is similar. So now out of 10 residues, how many are similar? There are 1, 2, 3, 4, 5. 5 similar. So what is the similarity percentage? So you can say what is the percentage of sequence similarity? Sequence similarity is out of 10 nucleotides, 4 of them, 1, 2, 3, 4, 5. Sorry, 5 of them are similar. So 5 divided by 10, and since it's percent, 100. So that comes out to be how much? Yes. What does this mean? That these two sequences are 50% similar. So sequence similarity essentially is a direct result of observation. What you did, you just wrote the two sequences and you aligned it, right? And you've seen how many were similar. Based on that, you could calculate the percentage. So sequence similarity can be a result of direct observation and can be quantitated in terms of percentage. Now, if I say the sequence 1 and sequence 2 are 50% similar, can I also say that they are 50% homologous? Can I say sequence 1 and 2 show 50% homology? No, that is absolutely insane. Why is it so? Because see, your DNA has just 4 bases A, T, G, and C, right? So there is a very good chance that this 50% similarity is just by chance. Just because there are 4, you got a similarity. Do you think that any given scenario that you can say that these two sequences are homologous? No, right? So sequence similarity and homology are not the same thing. So then how do you understand what homology is? So you can use the sequence similarity data to predict the homology. Means what? So sequence homology is an inference or a conclusion about common ancestral relationship. So whenever we say something is homologous, we say that maybe these two sequences or these two organisms have come from a common ancestor, right? So you can say two sequences are homologous after you look at the sequence similarity. So sequence similarity data can be compared to basically conclude if a particular sequence has a common ancestral relationship or not. Now, sequence homology is purely qualitative. So you cannot ever say that there is 50% homology. You can only say that it is homologous. If you have, say, a sequence similarity of 75%, you can say that the two sequences are homologous, right? We can never say it shows 70% homology because you don't know what the ancestral sequence is. So homology is a qualitative term, whereas sequence similarity is a quantitative term. Sequence similarity can be simply found out by alignment, whereas homology is a conclusion based on this alignment. Now, let me give you one example. What is one example? One example is you have one sequence that is, say, 10 base pairs, 10 nucleotide sequences long, and one is 50. Don't you think, depending upon the length, you will have different values of percent similarity? Like this is a very small sequence of 10 nucleotides. Therefore, you're getting a sequence similarity of 50%. Whereas if this was a 50 residue nucleotide sequence, you may not have got this much higher percentage, isn't it? So don't you think sequence similarity varies along with the length? Yes. So how do we then normalize this? So that can be done by using this graph. So how to say if a sequence shows homology or not. So whether it is homologous or not, how do we say? So that prediction can be done based on this graph of sequence length versus sequence identity. I hope you understood why we are talking about sequence length. Because the probability that a smaller length sequence will be homologous is high as compared to a larger sequence, because a random chance may be there since we have only 4 nucleotides. Therefore, to nullify this, to normalize it, we need some kind of standard. So we use this graph of sequence length versus sequence identity in percentage to find this out. Now you may ask, what is sequence identity? We studied about sequence similarity. I told you that is the total number of sequences that are matching, and divided by the total into 100 is the sequence similarity. Similarly, sequence identity can also be found. So to calculate sequence identity, this is the formula. Sequence identity I is equal to Li. Li stands for the length of identical matches. Like in our previous example, we found that there are 5 similar nucleotide, 5 matches. So Li would be 5 into 2 divided by La. La is length of sequence 1 and Lb is length of sequence 2 into 100. So by aligning and calculating the sequence identity, you can then determine whether it is homologous or not. So if after calculating the percent identity, you find that the percent identity is 30% or higher. 30% or higher means you can say that they have close homology, and you will say you're in the safe zone. So you can safely predict that these sequences are homologous if they have a 30% similarity or higher. If it is between 20 and 30%, you are in a twilight zone. Twilight zone is may be homologous or may not be homologous. Okay. And if you have a below 20% sequence identity, then you are in the midnight zone. That is definitely the two sequences are not homologous. To discuss some terms that are associated with these. So very frequently asked question. Please note down these terms. You can also have this slides of mine from my Telegram group. My Telegram group is right now very active, and I have very good people who are constantly answering on that group. So if you wish to join daily MCQs and avail all of these notes, please join my Telegram group. I have the link down in the description. Now, the first term that you have to understand is orthologs. So what are orthologs? Two homologous genes in different species. Remember, homologous genes in different species that have the same function are called as orthologs. So orthologs basically are homologous genes that have the same function. However, they are having different, they are present in different species. Then what are paralogs? Paralogs are when two genes, maybe in same or different species, have a different functions. So two homologous genes that are present in same or different species having different functions are called as paralog. Okay. So although they are homologous sequences, they do not perform the same function. They are paralogs. Orthologs, they may be of different species. Okay. The homologous genes may be of different species, but they perform same function. Paralogous, they may be of the same or the different species, having homology but completely different functions. One is the result of a convergent evolution, the other one is a result of a divergent evolution. Then you have heterologs. Heterologs are sequences that do not have any common origin. So that is the right opposite of a homolog. So heterolog is it doesn't have any common ancestor. Then we have analogs. So analogs, as the name suggests, they are analogous, meaning they have the same function, but they lack sufficient similarity to imply a common origin. So they perform same functions. However, you can't call them homologous because they do not have enough sequence similarity. This may be possible because both of these sequences may have evolved through different evolutionary pathways to reach to that point. Then we have xenologs. So xenologs, unlike the others, actually are an exception to the rule that homologs are from a common ancestor. Many a times the homologs might not have a common ancestor, and that is because the genes are passed via horizontal gene transfer and not in the traditional vertical transfer. We know that many organisms acquire sequences, acquire DNA from related species or from the environment, right? So those homologs which are resulting from HGT, that is, they are homologous but don't have any common ancestor, are called as xenologs. Hopefully, you understood this. Now, moving on to the next important topic, that is alignment scores and gap penalty. You have to understand this very, very well. So pay attention. Okay. Now, if I tell you to align this sequence, normally what you would have done is, okay, these are matching residues. So A matches with A and A matches with A. Now T doesn't match with C, so you will leave it. Then these two match. So only 5 matches are there. 5 matches. Now, how will you maximize the number of matches? Maximize the number of matches. How can you do that? Try. I will write the answer over here, but I want you to try. So the first sequence, I am keeping it the same, just taking this. A triple T, double A, G, G, G. Okay. Now, in the second sequence, what I'm observing is the first two residues were matching. So I keep them the same way. A, A. Okay, let's have the second sequence also, that is CT. Then you have GA, which are again matching. So till your stuff is okay. Now, see also, observe carefully, there is no match for these two T's over here, correct, in the second sequence. But if you observe, this CGC is present here, but there are these 3 nucleotides in the middle, these, that don't have any match. So in order to increase the matching, what I can do is, I can add something called as a gap. So I am adding a gap over here. That is, I'm leaving an empty space. Now, if I add a gap, After that, what I can do is I can align the rest. So after adding three gaps, where did I add these gaps? So we had added A, C, T, G, A. So A, C, T, G, A, then the three gaps. Now I can align this next sequence over here. That is C, G, C, A, T, then you have C, double T, double A, triple G. Now look at the alignment match. So let's count. So this is a match. Here you have matches. These are also a match. Now count the total number of matches. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. You have 16 matches now. So just by adding a gap, that is by leaving three places, you could maximize the match. So initially, if you wouldn't have added any gap, you would say that the sequence is hardly matching, right? But after adding a gap, you can see that the alignment score has increased and therefore the similarity in sequences can also be increased. But then you must be having a question. Okay, ma'am, can you just keep on adding gaps? So in that case, I can make my sequence 100% similar by just adding gaps. The answer to this is no. You cannot keep adding gaps. So then how to decide when to add a gap? And how can adding a gap be justified? So let me answer that question. Okay, how can adding a gap be justified, or where this concept of gap has come? So now during evolution, we know what happens is there are mutations that take place, right? And during these mutations, there are certain insertions and deletions that take place, right? So if you carefully look at the sequence 2, what might have happened is that these three nucleotides from the sequence 1 might have got deleted, or maybe the if in the sequence 2 at this position there might have been these insertions, three nucleotide insertions, and therefore these sequences have come. Isn't it possible? Yes, definitely it is. But can we 100% tell whether three nucleotides were added or removed? No, you can't predict, because you do not know the ancestral sequence. You don't know what was the ancestor's DNA sequence, and therefore you cannot make out whether it's an insertion or a deletion. But this gap is added to account for any insertion or deletion that might have taken place. An addition of this insertion or deletion may have happened because of evolution. So therefore, you can add gaps, or we add gaps to maximize the similarity or the alignment. Now, these gaps are many a times referred to something called as indels. Indel stands for insertion deletion. So a gap represents an insertion or a deletion. Okay. Now, the first question that I had posed that you might have in your mind is, can we just keep adding gaps? That way we can make a sequence similar. So if you're adding gaps, you have to penalize that gap. Meaning, now you are adding a gap to increase the alignment. So you have to take care that you are not adding unnecessary gaps. And how is that possible? By keeping something called as a gap penalty. Okay, that is, if you are adding one gap, you should minus one from your total score. So that way, any wrong gap addition will cost you some kind of some kind of loss in your alignment score. Let us try to understand what does this mean. So let us consider our same sequence that we have just written down, where we had 16 matches. Let's calculate the alignment score for this. So pay attention to how to calculate some alignment scores. It's very simple. Let's say that a match is equal to 1. So if a residue matches, you will give it a score of 1. If there is a mismatch, 0. Okay. Why 0? Because if it's a mismatch, it is a mismatch. Why do we have to cut any marks? Because you're not adding or you're not deleting. You have to just check the alignment. So 0. And gap, because obviously if you're adding a gap, you have to account for that gap. Otherwise, we can just keep on adding gaps to maximize alignment score. So how many matches we have? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Okay. So match is equal to 16. So you have 16 points over here. How many mismatches you have? You have 1, 2, 3, 4, 5, 6, whatever mismatches. Just not accounting for it because you don't have anything to minus. Gap. How much gaps are there? So 1, 2, 3 gaps are added. So you will minus 3. So your alignment score comes out to be 30. As simple as this. So this is how alignment scores are calculated. Now you will say, who decides whether we give 1 mark for a match or half mark for a gap, etc. So this is decided by different software. Every software has its own value which it takes, either 1, 0 or -1. Okay, so that is done with a number of calculations. Now, how do people determine what should be the gap value? So how to penalize gaps? So you penalize gaps by keeping a gap penalty. Now, scientists use different methods for penalizing a gap. 3 most commonly used methods are the constant penalty, proportional penalty, and affine penalty. Now, what is constant penalty? As the name itself suggests, constant, meaning irrespective of the length of the gap. So in our previous example, we had added 3 gaps. So let it be 3 gaps, 10 gaps, 15 gaps, you always cut -1. That is the constant penalty which we used while calculating the other score. The other one is a proportional penalty. Now, what people started saying is that, see, when you insert one gap, okay, that is when you should be charged the most, okay, as compared to opening that gap. Meaning, see, when you change one residue, it is one nucleotide, it is bound to happen that the next two will change, right, because of an insertion or deletion. So whenever you're adding the first gap, that should be penalized more as compared to the other two. So that is taken into account in the affine penalty, whereas in the proportional penalty, it says that the length of the gap should be penalized. So when you have 3 gaps, you should have a -3. But if you have 7 gaps, then it should be some constant number multiplied by the value. So that is B into L. B is nothing but the value of the gap. So let's say we said that a gap will be charged -1. What is L here? L says the length of the gap. So say you have added 3 gaps only, then your score will be -3. But if you have added 7 gaps, then it will be -1 into 7 -7 gaps, that way. So depending upon the length, you will be penalized for adding a gap, that is proportional. And finally, a fine penalty. A fine penalty is gap opening plus gap extension penalty. I'm sorry, I think during my proportional penalty time, I explained a fine penalty. So a fine penalty is opening the gap and then extension. So opening the gap penalty is A, whereas for continuing the gap, it's B into L. So it takes into account both constant and proportional in a way. So just to sum it up, why is a gap added? Gap is added to maximize alignment. And why people add gaps? Because during evolution, there might be insertions or deletions. So to account for those, we add gaps. Now gaps obviously need to be penalized. You need to take a penalty. So what is the penalty? How to decide? There are different ways to decide. You can either have a constant penalty, proportional penalty, or a fine penalty. Constant penalty is where you fix a particular value that this much will be deducted irrespective of the length or how many times the gap has been added. Proportional penalty is when the value of the gap will be multiplied by the length of the gaps that are added. Higher number of gaps, higher length of the gap, then higher penalty you have to pay. So if you add 3 consecutive gaps, you pay less. Whereas if you add 7 gaps continuously, you will pay more. That way. Then affine penalty is the penalty for gap opening as well as gap extension, both together. Okay. So that's all for me for this particular video. In the next one, we will be doing the similarity searches for nucleic acids. So please stay tuned for the next video. That's it from me for today. I'll see you in my next one. Bye.